Introduction. We're gonna see about fungi, mycology, myx, which is mean by mushroom in the Greek word. Fungi, they are the eukaryotic protista, differ from bacteria and other prokaryotes. While speaking about the characteristics, the cell walls containing chitin, which gives the rigidity and support, and they also have the mannose and other polysaccharides. They are cytoplasmic membrane containing ergosterol. They possess two nuclei with the nuclear membrane and the paired chromosome. They divide asexually, sexually, or by both. They are unicellular or multicellular in nature. While speaking about the characteristics of fungi, they are the diverse form of a chemo or heterotrophs, which is greater than 90,000 known species. They are saprophytic in nature, which digest the dead organic matter. They are also parasites, which obtain the nutrients from the tissues of the organisms. Moles and mushrooms are multicellular in nature, while the yeast are unicellular in nature. How the fungus are nourished. The fungus, they all are chemoautotrophs. Absorption of nutrients by the powerful exoenzyme. They grow at the lower pH 5 than the bacteria. They grow in the high salt and the sugar condition. They metabolize the complex CH2O in like lignin and wood to wood rot. Now we can see about the mycology. The characteristics like they present as the saprophytes in soil, decaying plants and nature. They are eukaryotes. They are known before the bacteria by the botanist. The developing nation changing the trends like USA have the more serious and even the fatal diseases because of the microbes. Fungi. The diverse group of the heterotrophs, many are ecologically important saprophytes. They consume the dead and the decaying matter, as I said before, other than the parasites. They are multicellular, but the yeast are unicellular in nature. Most are aerobes or the facultative anaerobes. The cell walls are made up of chitin, which is a polysaccharide one. There are over 1 lakh fungal species identified, only about 100 of the human or the animal pathogens. Most human fungal infections are the nosocomial and occur in the immunocompromised individuals and the opportunistic infections. While speaking about the structure of fungi, yeast and mold have the different structural and the reproductive characteristics. They are unicellular nucleated round fungi, while the molds are multicellular filamentous fungi. Yeast reproduce by a process called the budding, while the molds reproduce as opposed to reproduce. Some yeast are the opportunistic pathogen in that they cause the disease in the immunocompromised individuals. They are used in the preparation in the variety of foods. While speaking about the predisposing factors of fungi, we can use the antibiotics and the steroids and the immunosuppressive condition. Fungal diseases. Mycosis, a fungal infection which is lesser than 100, can cause the human disease. Not highly contagious, the humans acquire from nature. Groups are based on the degree on the tissues involvement and the mode of entry. The cutaneous mycosis, dermatophytes, which are mostly present in the epidermis, hair and the nails. Contagious, they are direct or indirect contact. They secrete the keratinase that degrades the keratin. And the next one is the fungal morphology. Many pathogenic fungi are the dimorphic, forming the hyphae at the ambient temperature but the yeast at the body temperature. They are vegetative structure which are involved in the catabolism and the growth. Thallus in the molds and flesh fungi. The tubular filaments of the cells called the hyphae which is present, and the septate hyphae, the cross walls that divide them into the unicellular unit. Pores to allow the cytoplasm and the nuclei to pass. Cenocytic hyphae, there will be no septae, continuous cells with many nuclei. The cell wall is rigid, which contains chitin, manganese and the polysaccharides. The cytoplasm contains sterols. They contain two nuclei, paired chromosome. They divide by sexually, asexually or by both. They can have the specialized cells. As I said before, they are eukaryotic, which are non-vascular organism. They reproduce by means of spores, usually wind disseminated. They are both sexual and the asexual. Bisexual, they can do the process meiotic, while asexual, they are doing the mitotic process by spores. It's depending on the species and the conditions they all live in. They are typically non-motile, although a few chytids have the motile phase. Like plants, fungi have an alternation of a generation. Understanding the terms in mycology, the simplest unicellular budding yeast. They are tubular elongation thread-like structures are called hyphae. The, if it is a tangled mycoplasm, it is called mycelium. Molding produces the filamentous fungi. Septate aseptate, they grown up aerial mycelium grows into the media vegetative mycelium. Simplest fungus, they are unicellular budding yeast. Hyphae, the elongation of the apical cell produces a tubular thread-like structure called the hyphae. Mycelium, tangled mass of hyphae is called mycelium. Fungi producing the mycelia are called molds or the filamentous fungi. Hyphae may be septate or the aseptate. Vacuole, the cytoplasm less dense in the older parts. Nuclei, the hyphae is sinusoidal aseptate. Rough endoplasmic reticulum, cell wall, nucleus, cell membrane, and the Golgi apparatus can be present in here. While speaking about the classification, depending on the cell morphology, we can classify it into 4 types. The first one is yeast, yeast with fungi, molds, dimorphic fungi, yeast, which is a unicellular fungi which reproduce by budding. On culture, they produce the smooth, creamy colonies, Cryptococcus neoformans, capsulated yeast. Yeast-like fungi, they grow partially as yeast and partially as the elongated cells resembling the hyphae, which are called pseudohyphae. Example, Candida albicans. Molds or the filamentous fungi, they form through mycelia and reproduce by formation of different types of spores. They are vegetative or the aerial hyphae. Example, Rhizophus. Dimorphic fungi, they occur in 2 forms. One is mold and other by yeast. Mold, which is filamentous, and they grow in the soil at 25 degrees Celsius. Yeast, which grow in the host tissue at 37 degrees Celsius. Most fungi causing the systemic infections are the dimorphic one. Example, Histoplasma, Blastomyces, Penicillium, Sporotherix, which causes the Brazilianese, comes under the dimorphic fungi. While speaking about the reproduction in fungi, asexual and the sexual reproduction, the sexual reproduction can occur by the formation of zygospore, ascospore, or basidiophore. Asexual reproduction by budding or the fission, the asexual spores are formed on or in the specialized structure. They vary in size, shape, and the color, but these characteristics are constant for the particular species. Microconidia, they are small, single-celled. Macroconidia, they are large, single or the mini-celled. Systemic classification based on the sexual spore formation. Classified into 4 classes: zygomycetes, ascomycetes, basidiomycetes, deuteromycetes. Deuteromycetes, which is the fungi imperfect type. Zygomycetes, they are lower fungi, broad, non-septate hyphae, asexual spores, sporangiospores present within the swollen sac-like structure called the sporangium. Zygomyces, the sexual spores. Zygospore, a resting thick-walled cell in between the hyphae. Example, Rhizophus mucor. Ascomyces, they include both yeast and the filamentous fungi. Narrow septate hyphae. Asexual spores are called conidia, borne on the conidia form. Ascomycetes, sexual spores called the ascospore. They are present within the sac-like structure called ascus. Several asci may be seen within the fruiting body as seen in the Penicillium aspergillus. Each ascus has 4 to 8 ascospore. Basidiomycetes, sexual fusion results in the formation of a club-shaped organ called the base or basidium, which bears spores called the basidiospores. Deuteromycetes are the fungi imperfecti, the group of fungi whose sexual phases are not identified, grow as mold as well as yeast. Conidia, example, Candida, Cryptococcus. Vegetative structures of fungi. The first one is the arthrospore, formed by the segmentation and the condensation of the hyphae. Chlamydospores, thick-walled resting spores. C. albicans. Asexual spores, they are produced by the aerial hyphae which is adapted for the disposal. Progeny genetically identical to parent. Several types like conidiospore, they are not enclosed in the sac produced in the chain at the end of the conidiospore. There are several types. Sporangiospore within a sac or sporangium they can be found. The end of the sporangiospore is the sporangiospore. Sexual spores. There are three phases of development. One is the plasmogamy, karyogamy, and the meiosis. Plasmogamy, haploid nucleus of the donor cell penetrates the cytoplasm of the recipient cell. Karyogamy, the two nuclei fuse to form the diploid nucleus. Meiosis, diploid nucleus give rise to the haploid nuclei. Sexual spores, some which contains the positive and the negative, and the some may be recombinant. The sexual spores used to classify fungi into divisions. Superficial mycosis, two types. One is surface and the cutaneous mycosis, skin, hair and nails. Mild but chronic disease. Deep mycosis, two types, subcutaneous and the systemic mycosis, caused by soil saprophytes. Infection is accidental, range from the symptomatic infection to the fatal disease. Superficial surface mycosis live exclusively on the dead surface of the skin and its appendages. No contact with the living tissue, hence no inflammatory response. Tinea vesicolor, tinea nigra, and the pairdra. Superficial cutaneous mycosis, cornified layer of skin and its appendages, contact with the living tissue, hence the inflammatory or the allergic response is seen. Dermatophytes, which is skin, hair, and nail. Three genera, Trichophyton, Microsporum, and the Epidermophyton. Characteristics of fungi. Dimorphic fungi can exist as both multicellular fungi and yeast. There are many pathogenic species. Mold form the produces the aerial and the vegetative hyphae. Yeast form the reproduction by the budding. And the dimorphism in the pathogenic fungi typically depends on the temperature at 37 degrees Celsius, which is the yeast form, and 25 degrees Celsius, which is the mold form. The dimorphism in the non-pathogenic fungi may depend on the factors carbon dioxide concentration. Mycosis, superficial, cutaneous, subcutaneous, which is systemic, and the opportunistic. Deep mycosis, subcutaneous mycosis, systemic mycosis. Subcutaneous mycosis is mycotic mycetoma, chromoblastomycosis, sporotrichosis, rhinosporidiosis. Systemic mycosis, cryptocrosis, blastomycosis, ozidiomycosis, histoplasmosis. Candidiasis, they are caused by Candida species, forms a bridge between the superficial and the deep mycosis, as it can cause the cutaneous as well as the systemic infection. It can also cause the opportunistic infections. Opportunistic infection, patients with the debilitating disease alter the physiological state, mainly caused by the fungi, which are common lab contaminant on the culture media. Aspergillus, Penicillium, Mucorrhizopus produce serious and the fatal conditions. Useful properties of a fungi, they are used as a source of food, and they can also used in the fermentation process and the antibiotic production. Ergot from the claviceps, which is used to induce the uterine contraction, and the vaccines for the hepatitis B can be used as fungi. Diagnosis and treatment, they are grown in a medium that selects for the fungal growth at 25 degrees Celsius and 37 degrees Celsius. KOH preparation of the skin biopsies. Leaves only the fungal cells. Leaves only the fungal cells. Therapy. Ampicillin B or the ketanocols are toxic to humans. While speaking about the therapy, as I said before, the ampicillin B, these are toxic to humans. Program